Plants may not have nervous systems or muscular systems, but they are able to respond to both external signals like environmental cues and also internal signals about their own physiology. One example is the potato. It has some morphological adaptations for growing in darkness without light, and these are called etiolation. But once it's exposed to light, it wants to reverse those processes so that it can grow normally, and that process is called de-etiolation. But you only want to undergo de-etiolation once there's light present. You may recognize the potato on the left, which could be growing in your cupboard or in your basement or whatever, wherever you store your potatoes. But if you expose them to light, it's going to look something like this. And you don't want to do this if you don't have light present, because what's the point of leaves if you can't do photosynthesis? This is an example of a cell signaling process, and just like with animals, which we talked about in regards to the endocrine system, there are three stages: reception, transduction, and response. Reception means receiving the chemical signal or photoelectric signal or vibrational signal, whatever it is. Transduction means turning that signal into then some sort of response, which is the action that the cell makes. The classic example is a hormone, and a hormone is a signal, and it's received by a receptor if there's a match, right? The receptor has to have the appropriate shape to bind with the signal molecule. Once that happens, you have to find some way to turn this signal, right, into a reaction by the cell. And that process is called transduction. And then the cell response is going to be, maybe you turn on an enzyme that produces a certain product or causes a certain metabolic process to go forward. Or maybe you turn on a gene that causes the cell to produce a protein it didn't produce before. Reception requires receptors, and sometimes receptors can be for chemicals like hormones. In this case of de-etiolation, the receptor is a phytochrome, which is a molecule that detects light. And you have similar molecules that are sensitive to light in the back of your retinas. They're not phytochromes, but you have rods and cones with different molecules that have different ways of responding to light. If you're going to turn that light signal or that chemical signal into a response by the cell, you have to transduce that signal. And this sometimes involves second messengers, right? Which means messengers that are released inside the cell to cause a change. So the hormone, for instance, might be a first messenger. The second messenger would be something like calcium or cyclic GMP, which causes a change inside the cell. So our first signal is light. It's received by this phytochrome. And this phytochrome, in response to light, changes shape. And that changing shape activates a second messenger. Could be cyclic GMP, could be calcium. It could open up a channel when it changes shape to allow calcium in. But the second messenger may activate some sort of enzyme that can cause something to happen, right? It may turn on a gene that can cause something to be produced. But either way, it's going to cause some sort of metabolic or physical response inside the cell. De-etiolation specifically is going to activate enzymes that are going to supply precursor molecules for chlorophyll, which allows you to undergo photosynthesis, and it affects the levels of plant hormones that are going to regulate growth, that are going to allow the shoots and the root systems to grow. Plants, just like animals, use a lot of chemical communication between cells in their bodies, and these are called hormones in plants too. Chemical signals that modify or control a physiological process or an anatomical structure within a plant. Hormones were discovered due to research into a phenomenon called tropism. And tropism is growth toward or away from a particular stimulus. Shockingly, Charles Darwin also discovered tropism. He and his son conducted experiments on how it worked. And they observed that grass seedlings could only bend toward light if the tip of the coleoptile at the top of the plant was present. If they snipped it off and didn't replace it, then you couldn't bend toward light. Darwin and his son postulated that there must have been some sort of signal transmitted from the tip to the rest of the region that was growing. And it wasn't until the 20th century that this was demonstrated, right? So Peter Boyce and Jensen demonstrated that there was a little mobile chemical substance. And then Fritz Wendt extracted the messenger by modifying some other experiments. This is an example of the basic kinds of experiments that were conducted by Darwin and later biologists. So if you cut off the tip of the plant where the coleoptile is, you see a lack of growth, right? If you then cut off the tip, put it on top of an agar cube, and then put that cube on there, so no tip, but the cube is filled with whatever goo was in the coleoptile, then you see growth, right? Indicating that there might be some sort of chemical in the tip that even if you put it in the cube, right, as long as you have the stuff that was there, it can cause growth. And if you only put it on one side, you only have growth on one side, and growth on one side causes you to bend in the opposite direction, right? If my back was growing faster than my belly, I would curve this way, right? And if you just put the agar cube as a control without any of the goo from the coleoptile, you don't see growth either. So it must be something that was in the coleoptile there. Just like in humans, plant hormones are produced in pretty low concentrations, but you don't need large concentrations to cause a big change. And plants have hormones that control growth and development, and they affect both the division and the elongation and differentiation of cells. This is a nice list of hormones that you tend to find in plants. That's from chapter 39, and you can look at it there. It's a good study material, but we're going to go through these hormones kind of one by one to discuss them. The first hormone discovered was auxin, still very important, and it's actually a group of hormones that promote the elongation of the coleoptiles. That's the hormone that was causing the effect we saw in the experiments that we just looked at. The most common auxin is IAA, or indoleacetic acid. So usually when we talk about auxin right here, we'll be talking about IAA. And it is produced in the shoot tips. It then is transported down the stem and can modify growth as you move down. How, you might ask? How does auxin promote cell elongation? And the mechanism is that it stimulates proton pumps. And when I say proton, remember I'm talking about a hydrogen ion, because hydrogen is just one proton and one electron. And if you get rid of the electron to make an ion, it's just a proton, right? We talked about that in Bio 131. And if you pump protons into a certain area, that means that area will have a higher concentration of hydrogen ions, and that means a lower pH. It's more acidic. And that activates expansins. And expansins are enzymes that loosen up a cell wall. And once that cellulose in the cell wall is loosened, it allows the cell to elongate. So auxin, the hormone, stimulates proton pumps. Proton pumps pump hydrogen ions into the area by the cell wall. That causes this to be more acidic, and that acidic environment activates expansins, which loosen up these cell walls and allow the cell to go from this length to this length. It allows cells to get longer. And there are multiple ways that you can grow, right? If you're going to grow as a plant, you can grow by making new cells or by elongating the cells you have. Auxin tends to work by elongating the cells that you have. Now that we know what auxins are and what they do, they're useful in agriculture. Some auxins can stimulate the growth of roots or the growth of cuttings of plants, so vegetative cloning. And too much auxin can kill plants, and so they're sometimes used as herbicides. Another example of a hormone is cytokinin, found in plants. And it's called cytokinin because it stimulates cytokinesis, or cell division. So auxin tends to stimulate the lengthening or growth of cells. Cytokinesis, or the division of cells, is stimulated by cytokinins. Cytokinins tend to be produced in actively growing tissues. So roots, shoots, embryos, fruits are also actively growing, and they tend to work with auxins to stimulate growth and differentiation. Auxin also works with cytokinins and strigolactone in order to maintain something called apical dominance. And this means that as long as there's an apical bud present, using these hormones, it will suppress the ability of axillary buds to grow. So the axillary buds are there, but they won't continue to grow as long as you have your terminal bud. But if you remove the terminal bud, then you stop making those hormones, the axillary buds are able to grow, and your plant essentially becomes bushier. So here are your axillary buds, one on this side and one on this side, and they are there, but they're not growing because you've got auxins coming down from above. If you remove this bud, then these axillary buds will grow and you get bushier. But if you remove this bud and then add auxin directly to the decapitated stem, it continues to suppress the growth of these axillary buds. So this is a kind of photo evidence from a study that shows that auxin functions in this apical dominance. Another important set of hormones in plants are gibberellins, and they have multiple effects that are important for agriculture, things like stem elongation, fruit growth, seed germination. Gibberellins are produced naturally in young roots and in leaves, and they stimulate the growth of those leaves, and then in the stems. And then in the stems, they stimulate an elongation and cell division. As you can see in the picture on the left, they can be used to stimulate a lot of stem growth quickly, and that does occur in agriculture. And they can also cause fruits to grow larger more quickly. And so I don't know if you remember when you were a kid, grapes were not nearly so large as they are now. Now you go to Kroger and the grapes are enormous. That's because they're treated with gibberellins before they finish growing and before they're picked and sold. Within seeds, embryos also tend to release gibberellins in the presence of water, and they stimulate germination, so growth from the seed. So again, GA, these are gibberellins. They're only produced in the presence of water, and that's so that you only will germinate under the right conditions, but that's the hormone that mediates that process. Conversely, abscisic acid is a hormone that slows growth, and it has effects like seed dormancy instead of germination, which is also important, and drought tolerance. Dormancy is essential because if a seed germinates in the wrong conditions, right, maybe in a dry year or maybe during the winter, then that seed is going to die. And so ABA helps maintain that dormancy, and sometimes it's removed by heavy rain or light or prolonged cold. Some plants need a long enough cold snap before in the spring they can germinate, and this is mediated by abscisic acid, which again, when removed, allows for germination. They also help in drought tolerance. For instance, an accumulation of ABA can cause the stomata to close pretty quickly, which means that you're not going to have as much water loss through those stomata through evaporation as you would normally. The last hormone I want to mention is ethylene, and it's produced in response to several different stresses, things like drought or flooding or mechanical pressure, so like pushing on a plant, or injury or infection, and it can cause some stress responses. It can also cause senescence or leaf abscission, so leaf dropping is caused by ethylene, and also fruit ripening. In terms of mechanical stress, this stimulates something called a triple response to avoid obstacles while a shoot or root is growing. So if you're growing up and you bump into something, it causes you to bend and then go around it until you can go back up. So you grow horizontally by thickening the stem until you can go north again. This is what happens to a stem in the presence of more and more and more ethylene, which again is secreted if you bump into something like a rock or something keeping you from growing and reaching the sunlight. Ethylene and its ratio to auxin also controls leaf abscission, which is what happens in the fall. It causes you to form an abscission layer and sort of block off the leaf from the rest of the tree, and that allows it to lose its chlorophyll and then die and fall. And it also allows fruit to ripen at the same time. Ripening fruit produces ethylene, which stimulates other fruit to ripen, and this is why if you want your bananas to get riper, you put them in a paper bag. At least that's what my grandma always told me, and that she didn't know this, but that traps the ethylene in the bag, ups the ethylene concentration, and it causes your bananas to become ripe more quickly.